Today, I'm going to be talking about two big problems that we have in single-cell omic analyses. The first problem is that we only have a single measurement for each cell. Now, this looks like the transcriptomic vector in single-cell RNA-seq that corresponds to that cell's transcriptome measurement. Now, this is comprised of essentially two major sources of effects. We have biological sources of variation as well as technical sources of variation. Now, one of the biggest sources of the technical variation is how deeply that cell got sequenced. That's introducing some level of Poisson sampling noise. Now, we have the ground truth transcriptome inside that cell, but we don't really know what it is. We're only able to see visibly some of the transcripts that get captured, successfully reverse transcribed into cDNA, and make it all the way through the processing steps to our final count matrix. Now, some of that signal is attributable to the noise in that sampling process. That means that if you have a sphere in similarity space, or hypersphere because we're dealing in greater than 3 dimensions, you can split it in half by doing k=2 clustering, and you can look at those centroids of the two cells, of the two populations that your clustering algorithm split it into, and those centroids, by definition, differ from each other, because that is part of how the clustering algorithm works. It seeks to maximize the difference between your cluster groupings. And so what this means is that no matter what you do, your subpopulations will always look different from each other, even if it's just Poisson sampling noise. Now, this was in part what they pointed out in Anna Neufeld's work looking at latent variable estimation for single-cell RNA-seq in combination with doing inference like differential gene expression. No matter what happens, because your clustering algorithm split a single spherical type of object into multiple parts, their centroids differ by definition because that's how the clustering algorithms work. Now, another problem that we have is even trying to figure out what the best ways to analyze your data is. Our benchmarking needs its own benchmarking in a way, because what we need to do is target how do we figure out what the most accurate methods are for our analysis. But to do that, we have to know the ground truth. Now, how do you know the ground truth in an experimental design like this? Now, there are some situations where we know. There's simulation and cell lines, but these, to be honest, are relatively trivial systems, and they don't truly represent what those of us who've analyzed, real-world data sets, they don't look like these simulations, they don't look like cell line data. They're not nearly that clean. So when we want to look at how different methods and pipelines perform in a complex data situation, what people will often do is take prior publications, take their published cluster labels, and then look at those labels as if they were ground truth. Now, in reality, what those ground truth labels are is just someone exactly like me who's doing their best applying status quo standard methods to analyze their data and then report those clustering results. Now, what this is essentially a measure of consistency with oftentimes the most popular pipelines. So this sets up an incentive where we end up benchmarking actually for precisely matching what's most commonly done in the field, which is wildly different than benchmarking for accuracy. So back to the first problem, overclustering in a way that is overfitting to that Poisson sampling noise. When you do clustering, you essentially build up your observations into a topology, and then oftentimes people will assess for differentially expressed genes between their clusters. And as I mentioned before, Anna Neufeld, working with Daniela Witten, pointed out that there's an issue here. And what that is the way that this works is you do cell-to-cell similarity calculations based on gene expression. Then you apply a clustering algorithm that finds parts of the data, a partition, that will maximize differences in gene expression. And then we check for differences in gene expression, which is logically circular, because that's how the clustering algorithm works. You can have a homogeneous looking sphere or hypersphere in n-dimensional space, and you'll always find differences. This is what accounts for the inflation of our significance values in single-cell data. So one way in traditional machine learning that people usually apply for trying to prevent overfitting in this fashion is to split their total data set into 3 data sets where we have training, validation, and test sets. And there's a problem with trying to do this approach with our situation, because if you generate cluster labels on your training set, you can't do differential expression on your test set because the cells that existed in your test set weren't in the training set, so they don't have cluster labels. And even if you try to do label transfer, that's only pigeonholing your test set into the training set topology, so there's still data leakage there. So I'm proposing some modifications that is totally informed by Anna Neufeld and Daniela Witten's work, where they proposed count splitting as an alternative way to get these different splits. So in this way, we have each cell appearing in the training set, the validation set, and the test set. And so if we picture a cell as a bag of marbles or a bag of transcripts, the way that count splitting works is you segregate your transcripts into different splits that are mutually exclusive, non-overlapping splits. So we have a bag of transcripts for training, a bag of transcripts for validation, and a bag of transcripts for test. And if we add them all up, we get our original observation. And so what does this look like when there's no structure present? What we want is basically a way where we can account for that Poisson sampling noise using our independent splits, because every cell has a measurement for our training, validation, and test. So as a first negative control, I looked at shuffled transcriptomes in HEC293T cells. Now, this is something that we know it's a hypersphere. It's the dimension of the transcriptome, but it's all shuffled up, so there's no genuine structure there. And if we look at the training set topology, there's this nice elongated continuum. And here it's spring embedding that's just colorized based off of the location in the topology generated from the training set. But if we look at the validation set, overall that topology structure looks very similar, a nice elongated strip like that. But if you actually look at the colors in the validation set, they're completely re-randomized. And if we look at an animation, it shows it a little bit more clearly that, yes, we end up seeing this nice elongated transcriptome or trajectory, but if you look at them in the training set and the validation set, they're completely re-randomized, because this method of count splitting is actually able to accurately quantify the consistency that's attributable or not attributable simply to the Poisson sampling noise. And so instead of looking at the, by definition, different halves of a sphere, the training labels are completely re-randomized in the validation set topology. So the centroids are now completely overlapping, and distributionally they look equivalent to each other, which is a good thing. That means that we can actually use this property as a way to account for that Poisson sampling noise. So what about when structure is present? Well, this is what the HEC293T transcriptome looks like in that situation. And in the validation set, we see that there is actually some concordance there. So our green cluster over there looks actually strikingly concordant on both sides of the map. But if we look at the blue and purple clusters, those tend to overlap, meaning that that elongated strip of the topology is actually, again, just attributable to the Poisson sampling noise and is non-reproducible between the training and validation set. So What does that look like in real numbers? Well, this is the actual data from that data set, where in gray, you're looking at the within cluster distances, and in red, you're looking at the distances between clusters. And you can see if we just look at the training distances, that's that self-fulfilling prophecy scenario, where we have, by definition, those centroids are separated. But if we use the validation distances, we can see that they're actually completely overlapping with the within cluster distances. So this is what the cluster validation algorithm that I'm proposing is built on, is using that independent validation split to validate the clustering results that we find in the training distances. Now, nothing in single cell is simple. One of the non-simple things about single-cell analyses is that we have continuous topologies. So there's just a schematic shape where each domain that is biologically, meaningfully, and reproducibly different from each other is in a different color. And let's entertain the hypothetical that our clustering algorithm split this topology into 6 different clusters. Now, we decided that one way that we could go about modeling this is a graph network. So each of these edges that are connecting the centroids to each other, those nodes in the graph, are edges of non-significance. So we connect them together if they're not significantly different from each other after Benjamini-Hochberg correction. And so now we have a graph representation of our non-significance within our data set based on the validation distances being non-concordant with the training distances. So our algorithm iteratively identifies the fully connected cliques, like this one, and then merges them into a single cluster, and then removes it from the graph. And now those 2 arms on the sides are no longer contiguous with each other. So our final answer is that we have 3 clusters. So how do we assess performance of this new cluster validation algorithm? Well, a simple start is using the simplistic data sets that we have, like cell lines, where we can use the shuffled transcriptome, but we know for sure there is absolutely no structure. The answer there should always be 1 cluster. And on the other hand, we can use the original cell line transcriptomes. And what we can guess here is that there should really only be 1 round of clustering. So we know in a cell line system that's homogeneous, not treated with anything, that there should be basically 1 level of variation. Now, there's going to be variation, as we've seen before, but there probably shouldn't be subclusters. So that's another heuristic that we can use for benchmarking. But again, I don't really even like this solution because it's hyper simplistic and it really looks nothing like a real-world single-cell RNA-seq dataset. So how can we actually assess performance in complex real-world datasets? As I mentioned before at the top, many approaches will use prior cluster labels as benchmarks, benchmarking for similarity to those prior labels. So let's say we have a single data set that's published with labels, and then we have pipelines that we're benchmarking against each other to see which of them can reproduce those labels with something like adjusted Rand index. But again, that's different from actually knowing the ground truth. And this is single cell omics data. The data is changing every day. New algorithms, new ways of analyzing things. Nobody's perfect, and I'll admit I'm the first one to say that I don't know the ground truth. And I don't think it's a good thing to necessarily just try to set up a benchmark to incentivize similarity to what's been done before. And one of the reasons for that is because if you look at different data sets like atlases, this is an example of an area in a ureter data set where there were B cells that came out in that data set, and you'll see that there are some circles in here that are highlighted in black around the edges. Those are samples from one particular batch that looked a little bit fishy, because if we look at not a marker for B cell identity in the cluster labeled B cells, but instead we look at a cell marker for urothelial cells, we see that this batch ended up getting overmerged, erasing the meaningful cell type variation. Now, if we're benchmarking using these labels as ground truth, we're just incentivizing to make the same mistakes again, rather than incentivizing for accuracy. So what do? If we can't use the original group's pipeline's labels, and we don't know the ground truth, what are we left with? Let's take a step back and ask the question, what is the point of clustering? So what I came up with is the ability to identify the greatest number of subsets of the data that are reproducibly different from each other. And that reproducibly different from each other gave me an idea related to count splitting. So we have already segregated out the training and validation set for the validation algorithm, where we can have higher confidence in our clustering results. But how can we use that test set? Well, if we're looking for reproducible differences and we have independent measurements of the same cell, then we can look at the test set to test the veracity of the clusters that came out from training and validation. And the way that we do that is by differential expression, because there are other ways of doing it, but we chose to use differential expression just for interpretability purposes. This is typically how people interpret their clusters, is by doing differential expression and looking at marker genes and all that sort of stuff. So the way that this benchmark works is by taking your first round of clustering, and we know from the impossibility of clustering theorem that may not be enough, but we do our first round, and then because no clustering algorithm or processing algorithm can perfectly identify all clusters, and what is a cluster is even subjective because distance is relative, there can be problems in doing a single round of clustering. So what we do is we allow these algorithms to recursively subcluster. And we do that for every cluster found in the first round and subsequent rounds all the way down. And then we look for whether or not inside of that branch there were no differentially expressed genes. If there are no differentially expressed genes that distinguish those groups in the test set, then we can conclude that those were false positives. And we do that within each branch of the recursive clustering tree. And then we allocate the false positives to the side. We identify what's called the Steiner tree of the significant differences, or significant partitions that did have differences between them. And then as a last sanity check, we check across branches to make sure that even across branches there are at least 10 up and down differentially expressed genes after correction. And you would be surprised, shocked at how frequently that happens, that we end up with across-branch false positives. So now we have an unbiased way to let the algorithms completely on their own identify true positives and false positives. And we won't get into the details for the sake of this talk, but I also found a way, when you're comparing a bunch of pipelines, to identify relative true negatives and relative false negatives, which opens up the full confusion matrix and all machine learning standard classification metrics. So by following this protocol, we've developed the first fully self-supervised benchmark for single-cell RNA-seq clustering pipelines. And this requires absolutely no knowledge of the system. You don't have to pretend like you know the right answer for the clusters. You don't even have to pretend you know the right marker genes, because we all have blind spots based off of the published literature or what we're expecting. So this has absolutely no prior expectation in it and allows for an objective benchmark. So this is the overall schematic of our benchmark, where we have the cell lines as well as the three complex real-world data sets. And ultimately, all of those differential expression tests added up to 100 billion differentially expressed gene analyses, or if we want to get grandiose with it, 0.1 trillion. And that is what led into all of these standard machine learning metrics from both sides, totaling 37 total metrics. We benchmarked 120 pipelines, head-to-head in this self-supervised benchmark where it's impossible for me to determine what the right answer is. It's an impossible to cheat benchmark, so we just let it go. And when we look at the results, displayed here as a heat map, looking at the rank performance for each of these metrics, and then sorted from least to most performant. And then the first thing we'll look at here is the feature selection method, colorized along the top. And then with and without cluster validation algorithm, this was the algorithm I described at the beginning. And then also looking at different distance methods, which I think there's a lot of room to dig in here more deeply. And we also looked at 2 different clustering methods, as well as different training to validation ratios. And overall, these are the Shapley values for the attribution of the most impactful features. So we built an XGBoost regressor of the average performance across all of those 37 different metrics, and these were the attributes of a clustering pipeline that most contributed to the highest overall average rank performance. And these 3 you'll take note of will come up again shortly. So one question is this new way of doing a benchmark where it's completely self-supervised actually giving us something biologically meaningful? It's a harder question to answer because then we have to go back to using prior expectation. So one way that we can do that, though, is by looking at previously described marker genes and seeing how much entropy each cluster has for those marker genes. So this is a semi-supervised benchmark, where we're taking our prior expectation of which marker genes should be important for each tissue, and then seeing, are our clusters giving us structure based on those marker genes? So how entropically distributed are our known markers within a cluster? Also normalized for the number of clusters. And so if we look at the scatter plot on the x-axis, looking at the self-supervised benchmark performance, and then on the y-axis, this semi-supervised known marker gene entropy-based performance. And the correlation was rather surprising to me how strong it was. Obviously highly significant. And if we look again at those Shapley values that came out, the anti-correlation based feature selection, which is an algorithm that I just published, was again at the top, along with application of the cluster validation algorithm, as well as this one, I will admit, surprised me. I was expecting the other distance metric to win, but reduction down to the first 50 principal components into the Euclidean distance was actually the best performing method in both the marker gene entropy approach as well as the self-supervised benchmark approach. So we can see here that the self-supervised benchmark clearly incentivized discovery of biologically meaningful groups, as validated by the fact that the groups that are prioritized, or the rankings that are prioritized by the self-supervised benchmark, match prior expectation in the form of entropy of the marker genes. So how meaningful are they? What can we figure out from this? So I took the best performing pipeline and tested the hypothesis using the liver data set that we analyzed, whether or not the true positive hepatocyte clusters are spatially variant. And so we first assessed the percentage of genes of the top marker genes from each of those clusters that were spatially variant, looking at the top 50 marker genes from each cluster versus 50 random genes. And we did this assessment using MaxSpin, which uses information theory. And yes, the top performing pipeline did have marker genes for the hepatocyte clusters that were significantly more structured and spatially variant than you would expect from a random set of genes that were expressed. And so if we just look at these hepatocyte clusters, and then look at the known zonation markers, we actually see that this spring embedding captures surprisingly well the orientation of the zonation patterns. So hepatocytes will vary in their cell identity based on their proximity to a central vein or a portal vein in the liver. And so we can see based on this zonation pattern that even just the spring embedding, which has a lot of caveats to it, and the distances are not completely reflective of the actual distances, that aside, we can see though that this spring embedding nonetheless was still able to capture the variation along the zonation patterns based on marker genes that are previously described. And so we were using the Visium dataset that was on the previous slide also for this analysis, and we wanted to check, looking at the transcriptomes of each of our clusters, if we could deconvolute those back onto the spatial transcriptomic dataset and see if we can calculate a portal to central score, and it matches perfectly based on our findings of the marker genes that, yes, on the left side of the spring embedding are the portal, most portal-like cluster, and then on the right side of that spring embedding is the most central-like cluster, or clusters. And if we look at the metabolic programming, we actually didn't expect this to come out. We just did blind differential expression, feeding in all of those results to pathway analysis, and actually the only thing that came out was almost entirely metabolic differences that were previously described. So here on the x-axis of all these plots is that same portal to central score that I just showed, and then on the y-axis is the mean standardized pathway cluster z-score, basically just a normalized version of significance. And we can see all the previously described pathways that are known to vary based on this axis, including amino acid metabolism, metabolism of lipids, and even some Wnt signaling that's known to be higher in the central side, and that will come into play in a minute. That being said, the reason that I think that's important is because we also fed these differentially expressed genes into survival analysis, and we ended up finding that central score seems to be associated with longer survival, which we think is associated with beta-catenin-driven hepatocellular carcinomas being slower to grow, despite the fact that they're actually harder to treat in some ways. So overall, we've used count splitting to enable independent topology validation, and I have an open source repository for this. And it's already freely available, pip installable, nice little README on Read the Docs and GitHub. And we've also shown that you can use count splitting to enable self-supervised benchmarking. You don't have to know the ground truth anymore. You can just let the pipeline decide what is the algorithm that gives me the greatest number of reproducibly different clusters. And the source code for this is also already available on Bitbucket. And one last thing, I've also released the count splitting, a Python implementation of the count splitting approach, which is now published, and big thanks to Anna Neufeld. And you should check out her R implementation if you're an R person. But yeah, I could not thank Anna enough for her kind words, guidance, and everything. She gave me some notes on my repository and the approach that I was implementing. Very helpful. Thank you, Anna. And I'd also like to acknowledge my collaborators on this work, my prior and current funding. Thank you.